see our products here. Okay. Another way of doing this, now it turns out that trying to identify these reaction intermediates is hard to do. Okay, those are difficult experiments. But anyway, another way we could do this, and this is just on paper, so you can do anything on paper. So on paper, what you can do is okay, you can draw the structure of each reactant and product and figure out, okay, how many bonds are forming and breaking in that reaction step. Okay, so if there's a lot of bonds forming and breaking, maybe that's not as probable as when there's fewer bonds breaking and forming. In other words, if we have a mechanism which shows one bond breaking and one bond forming, that may be more likely than this mechanism step which shows, say, 4 bonds breaking and 4 bonds forming at the same time. Okay, so that's the other thing you can do in that case. Okay, so going back to the iodide clock reaction, we know what the rate law for this reaction is, so we can propose a mechanism for this reaction too. So we'll spend some time doing that in lab today. Try to figure out what that mechanism for this reaction could be. Okay, let's talk a little bit about catalysts. Catalysts are very important. Note that I put on the bottom of the slide here, since you guys can decide, you can make a lot of money developing a good catalyst. So a catalyst does the following. Here's the properties: increases the rate of reaction. If it's called an inhibitor, it actually decreases the reaction rate. A catalyst also lowers the activation energy of a reaction. The catalyst is involved in a reaction mechanism. Okay, so it actually, what it does, it actually changes the reaction mechanism for a reaction. A catalyst is also regenerated at the end of a reaction. That's why you only need a small amount of catalyst. And then you should be able to use it over and over again. Remember that video from Chem1A a long time ago? They had this catalyst which cost like a few grams cost, they were saying $250,000. So they developed this catalyst, but since you can use it over and over again, although it costs a lot of money to develop, you can recycle it all the time to get it back. And then the catalyst is not involved in the overall reaction. So in other words, when you look at the overall reaction, you don't show the catalyst as a reactant or product in that reaction. Okay, so catalysts are found in our body. Those are called enzymes. The catalytic converter in our car is a catalyst. And then it's also found in many industrial processes. For example, to make plastics, a catalyst involved. To make ammonia, it's the Haber process involved. To make basically practically every industrial process to make some chemical involves some sort of catalyst. Now, the special thing about enzymes is that these are very specific for a reaction. So that's the hot part, that's the hot topic of research in biology now. Since the human genome has been sequenced, the next thing is to figure out what gene produces which protein and what's the function of that protein. Okay, so that's going to be a lot of work is being done now, and it's going to be done for many years on proteomics. Okay. Here's another one. I don't think we're going to finish this, but anyway, I'll go over this quickly. We'll go over this again next week. Now, hydrogen peroxide is used as a disinfectant. Now, what happens when you put hydrogen peroxide on a cut? Yeah, it burns, but also you see bubbles. What are those bubbles? Yeah, oxygen. So when it turns out that hydrogen peroxide is toxic to us humans, so there is a enzyme. There's actually a few enzymes. One is the peroxidase enzyme in our blood, and that catalyzes the decomposition of hydrogen peroxide. So here's the overall reaction. You have hydrogen peroxide decomposing into water, and here's the O2 gas, which is the bubbles that we see. One catalyst which you can use is iodide ion. So when iodide ion is used, this is the rate law using iodide. So note that it's first order in H2O2, first order in I-. Here is the mechanism of this reaction. So note that in the first step you have one H2O2 react with I- to form water and IO-. In the second step, H2O2 reacts with IO- to produce water and iodide-. Note that when you look at the overall reaction, the iodides cancel out and we get our overall reaction here. However, note also that the iodide is involved in the reaction. Note also that iodide is regenerated at the end of the reaction. Of these two steps, which step is the rate determining step? Step 1 or step 2? And why is step 1 the rate determining step? Because if we look at the rate law for this first step, it matches the rate law from the experiment. Okay. So what we'll talk about on Tuesday in class, we'll look at the rate law without a catalyst, and we'll compare these mechanisms, and then we'll draw another reaction energy diagram to see how a catalyst changes the activation energy of a reaction. But in the last few minutes of class, let's practice doing some problem solving by taking your 6th quiz of this.